Good afternoon, everyone. We're gonna start in 5 minutes so that people can join. Okay. So I guess we can start. So what I planned, as the attendance is quite low, uh, one update, I have started uploading the lectures on YouTube channel. So including today's lecture, as soon as I am finished with uploading all the lectures, I'm gonna share the link with you guys for that playlist so that you can go and watch the videos anytime. In the meanwhile, I'm also trying to figure out how to upload these lectures on the Moodle platform. As soon as I'm able to figure it out, I'm gonna share the link for that also. So we're not gonna study anything new today, because as we might be wrapping up our protein part in this week or next week, I just wanted that we should see from where we started and where we are heading, because the moment we continue our classes, chances are that we are forgetting from where we started and what are our basics at the end of the day, right? Things might seem very complex sometimes, very difficult to remember sometimes, so it's better to look back and see why it was important, from where it all started. It helps us in actually learning and understanding the topic in a better way. Okay, so we're gonna look back to proteins section in this class, and we're gonna have a quick recap about the lipids in general in the next class. Okay, so we are starting with the protein part. So as you remember, we started our discussion with cell membrane, right? So what is a cell membrane? Cell membrane basically defines the periphery of the cell, separating its content from the surrounding. Now we are pretty well versed with this terminology, right? And cell membrane has majorly two components, protein and lipids. Okay, so what are the major functions of a cell membrane? So cell membranes serve as barrier and gatekeepers. They are semi-permeable, which means that some molecules can diffuse across the lipid bilayer but others cannot. Small hydrophobic molecules and gases like oxygen and carbon dioxide cross membranes rapidly. Small polar molecules such as water and ethanol can also pass through membranes, but they do so more slowly. On the other hand, cell membranes restrict diffusion of highly charged molecules such as ions and large molecules such as sugars and amino acids. I hope that you guys are now understanding why this statement is very obvious now. Correct? Because of the nature of the membrane, this property is asserted by the membrane on these kind of molecules. The passage of these molecules relies on specific transport proteins embedded in the membrane. So a brief history of the membrane structure, how it all started and how we ended up with the current model, of which briefly explains the membrane. So it started way before 1925, but a significant discovery, and through which the idea of membrane structure was based upon, it was pivoted in 1925 via Gödel and Grendel model. They were the first ones to experimentally evidence that the lipid is actually, or our membrane is actually, in bilayer form and it is not a monolayer. Okay, then next 10 years down the line, Daniel Davison model came into play. Okay, which basically said that, though as Gödel Grendel said that it is lipid bilayer, they said that because the transportation is not possible of small polar molecules across the membrane, there has to be some proteins, right? So they described the model as Pauci molecular model. They basically said that though lipid bilayer is there, and on the bilayer side, on the heads of the lipid molecules, proteins are present. Okay, then obviously you can see in the cartoon representation of the Pauci molecular model that it is not true, because if it is true, it won't be able to incorporate that why there are proteins in the interior of a cell membrane, because now we know that proteins are also present inside the cell membrane, not only present on the surface of the lipid bilayer. Okay, then in 1957, Robertson model, which is called unit membrane model, came into play, which basically said that it's a lipid bilayer and proteins are there. That's it. But as it also has certain flaws, so it is a static model, and it basically fails to appreciate either a dynamic aspects of membrane or the asymmetric nature of membrane structure. Okay, then in 1972, the latest model which we work upon, which we study, came by, and it was given by Singer and Nicholson. They proposed the fluid mosaic model of the membrane structure, and you are seeing the representation of it. But on the other hand, though it is the widely accepted model, it ignores the role of lipid domains or membrane domains, which are oftenly found in the membrane. So a modification was done on the fluid mosaic model by Kai Simons, who proposed a lipid draft model for membrane structure, which says that there are small micro domains in the membrane which are transient in nature, which are saturated with cholesterol and some type of phospholipid molecules and lipid molecules, which creates these small domains which are thicker in comparison to the remaining part of the membrane, and they actually bulges out of the membrane when we see it. Okay, then we actually saw some of the representative videos, so we're gonna go back on some of them, not necessarily every representative video I'm gonna share. So let's see what we studied there. Let me share the screen with you guys.
Cell membranes are structures of contradictions. These oily films are hundreds of times thinner than a strand of spider silk, yet strong enough to protect the delicate contents of life: the cell's watery cytoplasm, genetic material, organelles, and all the molecules it needs to survive. How does the membrane work, and where does that strength come from? First of all, it's tempting to think of a cell membrane like the tight skin of a balloon, but it's actually something much more complex. In reality, it's constantly in flux, shifting components back and forth to help the cell take in food, remove waste, let specific molecules in and out, communicate with other cells, gather information about the environment, and repair itself. The cell membrane gets this resilience, flexibility, and functionality by combining a variety of floating components in what biologists call a fluid mosaic. The primary component of the fluid mosaic is a simple molecule called a phospholipid. A phospholipid has a polar, electrically charged head, which attracts water, and a nonpolar tail, which repels it. They pair up tail to tail in a 2-layer sheet just 5 to 10 nanometers thick that extends all around the cell. The heads point in towards the cytoplasm and out towards the watery fluid external to the cell, with the lipid tails sandwiched in between. This bilayer, which at body temperature has the consistency of vegetable oil, is studded with other types of molecules, including proteins, carbohydrates, and cholesterol. Cholesterol keeps the membrane at the right fluidity. It also helps regulate communication between cells. Sometimes cells talk to each other by releasing and capturing chemicals and proteins. The release of proteins is easy, but the capture of them is more complicated. That happens through a process called endocytosis, in which sections of the membrane engulf substances and transport them into the cell as vesicles. Once the contents have been released, the vesicles are recycled and returned to the cell membrane. The most complex components of the fluid mosaic are proteins. One of their key jobs is to make sure that the right molecules get in and out of the cell. Non-polar molecules like oxygen, carbon dioxide, and certain vitamins can cross the phospholipid bilayer easily. But polar and charged molecules can't make it through the fatty inner layer. Transmembrane proteins stretch across the bilayer to create channels that allow specific molecules through, like sodium and potassium ions. Peripheral proteins floating in the inner face of the bilayer help anchor the membrane to the cell's interior scaffolding. Other proteins in cell membranes can help fuse two different bilayers.
Yeah, so you guys get the idea. As we have already talked about most of the things, I'm not gonna play complete videos. So I'm not gonna play complete videos. I hope you guys can see my screen. So there was some trouble sharing. So I'm not gonna show the complete videos because we have already covered all these. It is just for a kind of a revision and kind of a thing that you can connect the dots, because we have moved a lot in this particular topic, and I want you guys to remember from where we started. If you are able to connect the dots, that the basic units of protein, membrane, properties, it will be very easy for us to actually understand the functions associated with the membrane and properties of a membrane. Okay. So this is basically the representation of a fluid mosaic model. As you can see, it's a lipid bilayer in nature, and there are different types of proteins which are present inside the membrane. Okay, inside and outside. So if you look at the inside, we can see integral membrane proteins, we can see peripheral proteins, which are not directly present on the membrane, but they are somehow managed to insert themselves inside the membrane, but still not directly linked to the membrane, right? Then we have on the other side of the membrane, we have GPI anchored proteins, we have oligosaccharide chains of glyceroproteins and glycolipids. This is the one which represents that our cell is of different types. These can act as biomarkers, and they help us in identifying different cell types. So it's a clear this is a clear distinction between the outside of a cell membrane and inside of a cell membrane. Okay.
And there was some network issue, so I got up some late joining.
It's okay, Shreyash. We are actually discussing our protein part from the beginning itself. I just wanted this one only because I'm gonna wrap up the protein part, and hence it is just a revision and a summary class for this part of the lecture. So you are actually not missing on anything. Also, the information which I gave at the starting of the lecture was that I am uploading these lectures on the YouTube, and I'm gonna share the YouTube link with you guys soon. Maybe in the moment I'll be able to upload today's class, I'll share the playlist link with you.
Oh, okay, ma'am.
So what were the major functions of a membrane? So as I have already mentioned, membranes are not there just to act as a barrier, though they are there, which are separating cell internal environment from the outside, but they are not merely there as a barrier. They are functional, right? This is what fluid mosaic model has told us. So they are flexible, self-repairing, and selectively permeable. Their flexibility permits the shape changes that accompany cell growth and movement, such as amoeboid movement. With their ability to break and reseal, two membranes can fuse, as in exocytosis, or a single membrane enclosed compartment can undergo fission to yield two sealed compartments, as in endocytosis or cell division, without creating gross leaks through cellular surfaces. Now you will be able to understand this point in a better way, right? Because as we have covered how the intracellular trafficking takes place, how the membrane proteins get assembled, this point makes more sense now, right? So it also imparts in the functionality of the membrane that they have this ability to fuse, and it's flexible and it can move. So all these properties actually helping us in getting the perfect intracellular movement of small molecules, as well as the transportation of molecules across the cell membrane. So and their selective permeability allows them to serve as molecular gatekeepers. Even now this point makes more sense, right, in comparison to the earlier when we had not discussed about the ion channels, the concentration of small molecules across the membrane, the ATP pumps, the potassium pumps, right? So this makes more sense now, and you can relate more to these points and properties of a membrane. These membrane functions are accomplished primarily by a broad array of proteins and enzymes in and on membranes. At the cell surface, transporters move specific organic solutes and inorganic ions across the cell membrane. As we have studied, this was the property of a membrane transporters, right? Receptor senses extracellular signals. We have seen GPCRs. We have seen GPCRs in photoception. We have seen GPCRs in cystic fibrosis. We have seen GPCRs. We know GPCRs also work as odorant molecules. Can sense odorant molecules also, right, and trigger molecular changes in the cell. Now we know how they trigger the molecular changes, right? They are secondary messengers. This is how they actually amplify the signal which was coming from a single ligand and multiplying thousands and thousands of times downstream just by having different secondary messengers, right? Then ion channels mediate electrical signaling between cells. This we still need to discuss. I am sorry I forgot about this, that I said we're gonna discuss action potential. Maybe in the next class along with lipids revision we can discuss action potential. Okay. And adhesion molecules hold neighboring cells together. We have not touched upon this, but I'm certain that you guys are aware of this. So you know there is one basic characteristic of a cancer cell. What is that? Cancer is basically defined as uncontrolled cell division, right? But the first step in the progression of a cancer during the metastasis, it means that from the original place wherever the cancer is being originated, it is now transferring and getting transported and circulated to other parts of the body. This is how cancer spreads. So for this, a basic property of a cell is being altered, and that is adhesion. The movement a cell decides to spread to other parts of the body, it needs to get detached from the place wherever it is situated, right? Hence adhesion molecules which are present on the membrane also play a major role in cancer diagnostics. Okay. The relative proportions of protein and lipid vary with the type of membrane, reflecting the diversity of biological roles. For example, certain neurons have a myelin sheath, an extended plasma membrane that wraps around the cell many times and act as a passive electrical insulator. The myelin sheath consists primarily of lipids, good insulators, and in contrast, the plasma membrane of bacteria and the membrane of mitochondria and chloroplast, the sites of many enzyme catalyzed processes, contain more protein. Even this point is more relatable now, right? As since the beginning of the sessions, I'm putting this point forth that membranes are asymmetric in nature. So the question arises from where this asymmetry is coming from. As biological membrane has two major components, that is lipid and protein. This asymmetry comes from both the lower leaflet of a membrane as well as the upper leaflet. Even the concentration of protein vary between different cell types. Okay, as the example which is given here, a neuronal cell has more lipids in comparison to protein, but a cell organelle such as mitochondria or chloroplast, they have more proteins because they perform enzymatic functions. They are functionally very saturated, and hence we need proteins, because lipids in general, they do not perform any function. Correct? They are there as a major component of a membrane to provide the matter in which the proteins can be easily embedded. Correct? So the functionality of a membrane comes from the different types of proteins which are present in the membrane, and hence according to the requirement of a cell, these type of protein as well as concentration of proteins will vary from cell to cell, even from organ to organ, and even within the organ also. Okay. So within the cell, membranes organize cellular processes such as the synthesis of lipid and certain proteins and the energy transduction that produces ATP in mitochondria. Okay, so this was basically the brief about the membrane proteins. And as I have discussed even today about the fluid mosaic model, we need to understand why this model is very important and why it is still widely accepted model, right? There are no second doubts that membranes are not fluid mosaic in nature, right? So this was a very good video. I personally find this video very informative, and I really hope that by me by sharing this video with you guys, that it will be very much informative to you too also. So let me play this for you. I hope you guys
explore
can
the
see my
fluid
screen.
mosaic
No, you
model.
guys cannot see my screen. Yeah.
Of cell membranes. Now, why is it called the fluid mosaic model? Well, if we were to look at a cell membrane, and just to be clear what we're looking at, if this is a cell right over here, if this is a cell, and this is its membrane, this is the membrane, it's kind of what keeps the cell, the inside of the cell, separated from whatever is outside the cell. We're looking at a cross-section of its surface. We're looking at a cross-section of its surface. We're down here, this is inside the cell. If we look at it relative to this diagram, this is inside the cell and this is outside. This is outside. And when you zoom in, this little part right over here, this is actually a phospholipid bilayer that forms it. And so when you hear that, you might say, well, what is a phospholipid? And that's a good question, because when you understand what a phospholipid is, it starts to make sense why it would form a bilayer like this and why it's the basis for so many membranes and biological systems. So this is indicative of a phospholipid. And as its name implies, and let me write that down, this is a phospholipid. It's a lipid that involves a phosphate group. And in general, the word lipid, and we have a whole video on lipids, means something that doesn't dissolve so well in water. And that's true as the case of this phospholipid. You have these hydrocarbon tails that are coming from fatty acids. And so these hydrocarbon tails, they have no obvious charge or no obvious polarity. We know that water is a polar molecule. That's what gives us its hydrogen bonds, and it's attracted to itself. But these don't have those, and so they're not going to be attracted to the water. The water's not going to be attracted to them, and so these tails are hydrophobic. So you have hydrophobic tails. And these are really kind of the lipid part of the phospholipids. And then you have the phosphate head. You have the phosphate head right over here. And as you can clearly see, this has some charge. Charged molecules do well in polar substances like water. They're going to dissolve well. And so this part right over here is going to be hydrophilic. Hydrophilic. And actually, molecules that have a hydrophilic part and a hydrophobic part, there's a special word for them, amphipathic, a word that I sometimes have trouble saying. So phospholipids are amphipathic, which means that they have both a hydrophilic end, a part that is attracted to water, and a hydrophobic end that is not attracted to water. And hopefully that starts to explain why they organize themselves in this way. Because you can imagine the hydrophilic heads are going to want to be where the water is, which is going to be either outside the cell or inside the cells. And the tails are hydrophobic. The water is going to go away from them, or they're going to go away from the water. And so they're just going to face each other, and they're going to be on the inside of the membrane. But the really cool thing is a structure like this, having this amphipathic molecule, allows things like these lipid bilayers, I should say, to form. And it's actually fascinating if we think that if you go far back enough, even before life in cellular form formed, that you might have had phospholipids spontaneously forming these spheres where you have a bilayer, a lipid bilayer. So you can imagine something. Let me see if I drew a cross section. Let me see if I could draw it relatively neatly. So I think I'll draw half of it, just because I'll draw the whole thing. Hopefully you get the idea. So that would be one layer of the phosphate heads facing the outside. This is the inner layer. And I'm doing a cross section right over here. And then you have your hydrophobic tails. So your hydrophobic tails, let me do that in a different color. So your hydrophobic.
Right, so as I mentioned in the beginning of the session that I'm not going to play all the videos because this is just a revision. We have already discussed this. It is just there for you to remember everything which we have learned till now about the membrane proteins. Okay, so it's clear, right, why we need the fluidity in the membrane. Now you guys can understand it in a better way because now we have actually discussed the functionality of the proteins which are present in the membrane, and if the fluid property, the fluid nature of the lipid bilayer is not there, all this functionality will be at halt and nothing can easily pass from one side to the other side. Hence we actually need the fluid nature of the membrane. Okay, these are just basic points which we have already discussed. The type of lipids and the proteins which are present in the membrane are amphipathic. Why? Because if a integral membrane or transmembrane protein is present, it needs to face both the hydrophobic environment of the lipid membrane interior, as well as the polar environment which is outside the lipid membrane. Hence this protein should have both the type of nature of amino acids which can easily be adjusted in the hydrophobic environment as well as in the polar environment. Okay, then what exactly is fluid mosaic model? The fluid mosaic model explains various characteristics regarding the structure of functional cell membrane. According to this model, there is a lipid bilayer, right, in which protein molecules are embedded. So if someone asks you one line definition of fluid mosaic model, this is it, right? But if you want to give more details, then what kind of lipids are present, what kinds of protein are present, you can easily get into those nitty-gritties of a membrane model description. So a membrane, the components basically are phospholipids, carbohydrates, cholesterol and proteins. Now you have better image of it, right? We have discussed phospholipids which were of different types. We have not discussed carbohydrates, but you do know that carbohydrates are only present in the exterior of a cell membrane, like the outer leaflet of a lipid bilayer. Then cholesterol, it is embedded in the membrane and helps in maintaining the fluidity. And then proteins, they are the rock star of membrane, and hence they are widespread and perform different functions. So then we discuss some of the experimental evidences which actually hypothesized and help us in actually saying that membrane is fluidic in nature and proteins are actually present both inside as well as outside of the membrane. Then the question arises, if this structure is so important, right, and so how it is stabilized at the end of the day. So one of the very curious observation in biology is that however the macrostructure is there, most of the times it is being stabilized by non-covalent interactions, right? So one covalent interaction, definitely it's more strong, but if there are hundreds and hundreds of van der Waals interaction or non-covalent interactions, they can overcome the effect and the energy of a single covalent bond, right? Hence in membrane biology, membrane lipids in the aqueous environment are basically stabilized by 5 major forces. These forces are hydrophobic effect, caged tails, the interaction which is because of the lipid side chains, okay, then head-to-head interaction which are polar in nature, then head-water interaction which is again a kind of a polar interaction, and then van der Waals interaction. Okay, so as I was saying, though covalent bond has more energy, but if we compare with the number of covalent bond and the number of non-covalent bond interactions, it actually overpowers. Hence most of the macro structures in biology are actually stabilized by non-covalent interactions. Then we discussed about lipids. So lipid is derived from the Greek word lipos, meaning fat. Therefore lipids are by definition insoluble in water but soluble in non-polar organic solvents. This is basically chemistry 101, like dissolves like. Because of this property, as the lipid molecules are coming together and making this membrane, they are not allowing molecules which are polar in nature to cross through them, to pass through them. To assure that even such molecules can be transferred from inside to outside or outside to inside, we have the need of proteins, correct? So there are n number of functions for lipid molecules, as you can see, which are enlisted here, right? Though classifying lipids is a useless task, because we know there are thousands and thousands of lipids which are even not been identified till now in the biological system, right? But still we need to start from some place. So the classification of lipids can be done on the basis of non-hydrolyzable lipids, simple esters, complex lipids, glycolipids, right? Major type of lipids which are present in cell membrane, they belong to these categories, right, which is present in the biological system basically. So we have fatty acid, we have glycerolipids, we have glycerophospholipids, we have sphingolipids, sterols, prenols, saccharides, and polyketides. And these are just how we can say that the series of even numbered carbon saturated fatty acids from C2 to C12, C2 to C24 means carbon to a lipid molecule having 2 carbons to lipid molecule having 24 carbons with their melting point and their water solubility. You can clearly see as the number of side chain carbon or the tail increases in lipid which are completely saturated indicating there is no double or single bond, you can clearly see that the melting point is increasing. Okay. Then we discuss a lot about the structures of lipids. You can see phospholipids, how they are made, how we can check whether a phospholipid is present or not. Then different types of phospholipids are present in the cell membrane. Now we are being membrane specific. Till now we were biological system specific, now we are being membrane specific. So in membrane specific phospholipids we have phosphatidic acid, phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine, phosphatidyl'inositol, phosphatidylglycerol, and cardiolipins. This is the general representation of a phospholipid, in which this is the glycerol backbone, a phosphate head group which has different alcohol groups attached to it. Based upon this alcohol we have this many types of phospholipid in the membrane, and these are the acyl chains. It can be the saturated or unsaturated. Then we discussed about individual lipid molecules also. We discussed about phosphatidic acid, phosphatidylethanolamine, phosphatidylcholine, serine. And then we saw how within an organism how the number of lipids within a cell or within an organ varies, right? PCs, phosphatidylcholine, phosphatidylethanolamine. So even within a organ, the number of lipids changes from one cell to another cell. Hence they are very diverse in nature. Then we saw how basically these lipids are distributed among the cell and the organelles, and we can see there is a huge mismatch, and there is no one number of lipid or n number of lipid fits all these organelles, all the plasma membrane all together, because every cell, every organelle, every membrane has different function to do, and hence their lipid composition also varies, right? Then we talked, and now if you remember this PIP2 and PLC, we have talked about GPCR signaling, right? So this was one of a kind of a secondary messenger. So now you understand from where we have started and where we are heading, right? Then sphingolipids are another type of lipids which are present in the biological membrane. Depending upon the group, sphingolipids can be termed as ceramide, sphingomyelin, cerebrosides, globosides, and gangliosides. You guys don't need to worry about remembering or like keeping a track of these names, but you should know if I say cerebroside, so you should have a mental image of that cerebroside. Oh, it's a type of sphingolipid, right? And it's not a phospholipid. Then we have talked about sterols, and then we have seen how conserved sterols are from fungi to mammals and their structure. And then we have talked about lipid distribution. As I have said, even within an organ there will be different cell types, and different cell types have different lipid distribution within themselves. right? So this was more from lipid point of view, and this is an introductory lecture which we actually started since this course was started. I'm getting certain mails regarding revision classes, so that is why I have scheduled this. Overall, whatever we have discussed till now in today's class, and maybe in the next class, that is, it will be on Saturday, coming Saturday, we're going to discuss about the proteins, whatever we have discussed. So though I have just flown through most of the things, if you guys have time, please go through these slides. I'm uploading them on the YouTube. I'm definitely going to share the link as soon as possible. Then refer to the information wala videos which I have already linked in the slides itself. They actually help us in generating a concept and give us some visualization. It aids in more understanding and learning of the topic. Okay, so see you guys in the next class. Thank you.